My name is Kitzel Lusted. I'm an undergrad researcher working with Dr. Michael Free, and today I'll be presenting research that's looking to extract rare earth elements, or REEs, from coal waste in a cost-effective manner using biooxidation. REEs are heavily used metals that have applications in magnets, alloys, as well as electronic devices such as smartphones. Significant concentration of REEs have been found in coal waste, but they're usually chemically bound to constituent components. This project aims to isolate them using biooxidation. Before I begin, I'd like to thank the University of Utah and Dr. Free, as well as the Department of Energy, for providing funding for this project. Biooxidation is the process of using bacteria to extract a desired metal without compromising that metal. The bacteria that we use, Acidotheobacillus ferrooxidans, is not the only bacteria used in biooxidation, but it was the most cost-effective choice for our experiment. Its primary function is to reduce the iron 3 plus ion to the iron 2 plus ion, producing an iron precipitate which contains the desired REEs. In our reactor, seen on the left, pyrite is used as the main source of food. It's pumped into the reactor from this flask. And at the same time, the leachate solution from the bioreactor is collected in the large white container seen behind the bioreactor. One of the main factors that we found to affect the rate of biooxidation is the concentration of calcium in the pyrite feeding mixture, which negatively affects their productivity rate. How we monitor the success and vitality of the bioreactor is twofold. The EH is measured to monitor the biooxidation rate and is quantitatively expressed by a ferrous oxidation test performed weekly, which can be interpreted using Michaelis-Menten kinetics. The smooth trend in the ferrous ion concentration plot on the far right indicates that the bacteria on November 6th were able to consume the ferrous ions at a relatively fast pace of around 3 to 4 minutes. The pH is also measured to measure the bacterial acid production. A low pH of below 2 indicates that the bacteria are healthy and producing lots of sulfuric acid. A high pH of over 2.6 means that the bacteria are not consuming the pyrite and there is something wrong with the reactor and they are most likely not reproducing. Once the leachate solution has been collected, it's filtered with filter paper. Anything that does not pass through the paper is collected as a filtered cake, which then can be analyzed using several different methods. Analysis with EDS, as shown on the left, shows that this particular leachate solution had high concentrations of europium and praseodymium. In conclusion, the methods used are a viable way to cost-effectively extract REEs from coal waste. As seen in Figure 5, iron precipitates begin to form around a pH of 2. The main precipitate was found to be jarosite, which is an iron-rich compound. This is useful information because the pH of the reactor should be kept to below 2 pH to ensure that precipitates don't form inside the bioreactor and clog the tubes leading in and out. Of the total REE recovery, shown in Figure 6, yttrium was the most prevalent at 22%, with cerium and neodymium following at 21 and 14%, respectively. The future work of this project will aim to refine the biooxidation technique to ensure that it can be used on a large scale in a cost-effective manner. Thank you.